Good afternoon, Rishika. How are you doing? Okay, so in the last class we had discussed about the specificities regarding the cell cycle. I had explained to you the significance of cyclin, the CDKs, the cyclin dependent kinase inhibitors CKI, and also you can say the activators of CDKs, right? So CACs and CKIs we had talked about also. So today's class we'll be talking about, you can say, so cyclin dependent activating kinase CAC, CKI, then the cyclin itself. What they do is they increase or they activate in the end the CDKs, they cause the conformational change in the case of CDKs, and in the end the CDKs are bound to cyclin, and if they are also bound to the CACs, okay, then they become completely activated, and after being completely activated they can then cause phosphorylation. They can add phosphate group by hydrolyzing ATP, because the kinases, they do what? They'll get phosphate group from where? Like they can add the phosphate group, but from where will they get the phosphate group? What's the source of the phosphate group? So the source of the phosphate group is the ATP molecule, which is the adenosine triphosphate molecule. So I'll just show the structure of the ATP to you. Just look here, ATP structure. Just look at this image. So this is the meaning of adenosine. Okay, adenine plus ribose sugar. That is a nucleoside that is also known as adenosine. And when you have only one phosphate, that becomes a nucleotide known as adenylate. But this is adenosine triphosphate, means we can call it a nucleoside triphosphate, means adenine plus the ribose sugar plus three phosphates attached at the same number carbon. Okay, so on the sixth carbon they are attached, right? So you can see here phosphate number 1, phosphate number 2, phosphate number 3. This is the alpha bond, right? This is the beta, this is the gamma phosphate bond. So this bond is broken down. Look at the gamma phosphate bond. This is broken down, and then this phosphate group. Okay, this phosphate group is then, which is which comes out as a result of hydrolysis of an ATP molecule. This phosphate pi or phosphate group is then attached, or then this enzyme kinase attaches this phosphate group to any, suppose, the tyrosine residue of any protein or serine threonine residues, any specific amino acid, okay, of that specific protein. This is how they operate. So now coming on to the next phase of the cell cycle, coming on to the, you can say, the S phase. Now you have entered the S phase of the cell cycle. Now what will happen in the S phase? Okay, so one more pathway I will tell you where you can go from, or where you can really activate the S phase in the cell cycle. Now one of the most important substrates, okay, of then the G1 S phase cyclin, okay, CDK complexes, that is DEAP. If you remember last time I had given you the specific details in a, you can say, tabulated way. So is CDH1. Now what happens is during, you can say, the S phase, okay, during the S phase what will happen is the phosphorylation of CDH1 occurs. Okay, there is a protein known as CDH1. Okay, so I'll just draw the diagram, it will become very easy for you. So what happens is, so there is a specific protein known as APC. Okay, APC slash C. Okay, this is known as anaphase promoting complex slash cyclosome. Now, this protein can block, can degrade the S phase cycling, but you can think of it as this protein cannot really recognize the S phase cyclins on its own. In order to recognize the S phase cyclin and you can say inhibit the S phase itself, means inhibit the replication of DNA in the S phase itself, block the cell cycle in the S phase itself, what it needs to do is it needs to attach itself to another protein known as CDH1. When APCC, this anaphase fermenting complex slash cyclosome, this protein is attached to CDH1, then it can recognize the S phase cyclins. Then it can definitely recognize the S phase cyclin and it will be able to then phosphorylate the S phase cyclin. Okay, and then what would happen is it would degrade the S phase cyclin. That's what it does. It causes the degradation of the S phase cyclin by a process known as ubiquitination, which is not in your course. So that is degraded. That S phase cyclins are degraded. Okay. So what happens is now this CDH1, now G1 S phase cyclins, they rise in their level. Okay, during the transition from G1 to S, now the G1 S phase cyclin, they rise in their level. Okay, now you must think of this thing as why is this inhibition necessary? That is also a thing of question, not that you need to revoke this inhibition just like that, but why is this inhibition even occurring? Okay, that means again a fundamental question in biology, like why are all these, you can say, inhibitors are there like E2F RB. So I told you if the inhibitor is not there, you can directly come to the S phase without your organelles being completely duplicated. So that will pose a great problem, a great, you can say, dilemma for the cell itself, right? So then what we can do is we can directly think of this event also as why it is happening, because biology is all about answering the why. Okay, going for the reason. The thing is, why are S phase cyclins being degraded? Now what happens is if they are not degraded, okay, so what happens is the APCCDH1, if it is absent, okay, suppose it is absent, what can happen? A cell can immaturely enter the, you can say, S phase or not. They can immaturely enter S phase, and immature entry into S phase means their organelles haven't been able to divide. Okay, their organelles haven't been able to duplicate and they have entered the S phase, and again their DNA is being duplicated but not duplicated two times. It can be 2.5 times, it can be 3 times. Again, that will cause chromosomal abnormality, aneuploidy, right? So those things can happen. So when the G1 phase is complete, E2FRB has been able to be, you can say, E2F RB has got dissociated. E2F is different now, RB is completely dissociated from E2F. Now E2F has commenced the G1 phase and the G1 phase has been able to see its completion. Now when the G1 phase has been completed, okay, now what will happen is now G1 S phase cyclins will rise in concentration. Okay, now when G1 S phase cyclin, G1 S phase cyclin, they rise in their concentration, so they will bind with the specific CDKs, okay, G1S cyclin CDK, and that will activate this CDK. Now when this CDK has been activated, okay, what will happen is it will cause phosphorylation of CDH1, just like phosphorylation of RB happened, and then RB was dissociated from the, you can say, E2F. Now RB, when it was associated with E2F, what we were looking at was E2F got completely repressed. It acted as a transcriptional repressor. And when RB got dissociated after its phosphorylation by G1 cyclin CDK complex, Then E2F was able to recruit the RNA polymerase and cause the progression in the G1 cycle. Now, same way here, when here it is different. APCC when attached to CDH1, it is very active. There E2F when it was attached to RB, it was repressor, but here it is active. In that case, G1 cyclin, G1 cyclin, CDK. Okay, what it did was it phosphorylated this one and this got dissociated. It became E2F plus RB. Now E2F was completely free. Okay, now free means it acted as an activator, and this was phosphorylated, so it could not be associated, it could not remain associated with E2F. Now here G1SCDK is active. This will phosphorylate at multiple protein residues, this CDH1. After this phosphorylation, okay, same way like RB got dissociated, it will get dissociated. Now CDH1, after it has been phosphorylated, it will get dissociated from APCC. When it gets dissociated from APCC, now APCC cannot recognize, now it cannot recognize the S phase cyclins. Thereby it cannot phosphorylate, cannot cause a degradation of the S phase cyclin. Now that is what is happening. Now you can enter the S phase properly. Okay. Now you can enter the S phase properly and can cause the DNA replication. Okay. Now after you have entered S phase, you may enter G2, you might have entered then the M phase, the mitosis phase. Now only as the mitotic CDKs, now you have entered the mitotic phase, suppose from S phase DNA replication done, G2 done, now have entered the mitotic phase. Now DEAB, now you are in the cyclin B CDK1 complex, now you are in the mitotic phase. So when the mitotic CDKs decline, what happens is that time a phosphatase protein known as CDC14 gets activated, okay, and then it will remove this phosphorylation and will again activate, so that you don't activate S phase again. So suppose this is the diagram, this is a clear cut diagram that I'll draw. APCC CDH1, it gets phosphorylated by G1 S phase CDK. Okay. So what will happen is you will get this as the result. One multiple phosphates could be attached. Okay. Now, when you have reached the mitotic phase, from here you have reached the S phase. Now S phase is active. Now, if the cell has reached mitosis phase, you can say M phase. At M phase, you can have a specific phosphatase activated, known as CDC14. Okay, phosphatase. Now, phosphatase will do what? It will break these phosphoanhydride covalent bonds. So it will make CDH1 dephosphorylated. These bonds will be broken. This covalent bonds will be broken. It will be dephosphorylated and it can again reattach with APCC. Now, why did it happen in mitosis? Is like after mitosis you enter G1 and not directly the S phase, because after mitosis the cell has divided. Now you don't need to divide the DNA. You don't need to replicate the DNA again. You need to first replicate the organelles. Okay, so think how great is your evolution, how great is our body, how enthralling is our body, how it is controlling the various pathways at correct time, at correct place, and in a correct manner. That you, once you have completed mitosis, you have got two cells. Now if it stays like this dissociated, now again the DNA will be doubled before the organelle duplication. So now then you have directly entered S phase, then again M phase, then how will the organelles duplicate? The cells will die after division. Then again it is deactivated, so that it will be like this. FPGC is associated with the CDH1 and it will stay, and it will start to, or it will commence to degrade the S phase cycling, so that it enters the G1 phase again, not directly the S phase. You don't want to directly enter the S phase after the cells have been divided after the mitosis event. Okay, so this is how the entire thing pans out, right? Regarding the S phase, G1 to S, then S2, you can say directly the M phase. Okay. So is there any doubt here, Ishika? Any kind of queries? Any queries here regarding the FPGC CDH1 pathway? Okay. Okay, good. So now we will be shifting our attention to, you can say, the activation of mitotic CDKs to initiate mitosis. Okay, to initiate mitosis. Okay, so mitotic cyclin CDKs, we know that they initiate mitosis, right? And we also know that the catalytic CDK subunit are constant throughout the cell cycle, and only the mitotic cyclins, they their level, it keeps on fluctuating. That's the golden rule in the cell cycle. Okay. So what happens is, what we have a control, or we have something known as a very great control over mitotic cyclin, okay, which is known as the G2 to M phase transition. Like when you have entered G2, you need to again, you can say, activate the mitotic cyclin, and then you can go to the, you can say, M phase or mitosis phase. Okay, so what happens is, there is a specific kinase, okay, a specific kinase which will phosphorylate, okay, the mitotic CDKs at specific amino acid residues. Okay, so there is a specific kinase. First kinase is known as V1. Okay, V1. So here from G2 to M transition, G2 to M transition, there is a specific protein kinase known as V1 kinase, means it will phosphorylate. Okay. Now V1 is a very specific kinase. Okay. So it can cause phosphorylation of two types of amino acids, or two other entirely different class of amino acids. One is tyrosine, okay, and one is threonine. It can cause phosphorylation, means it can add phosphate group of tyrosine and threonine also. Phosphorylation of threonine. Now, what happens is, it is also known as a dual specificity kinase. Why? Because there are two families of kinases, one which phosphorylate tyrosine, one which phosphorylates serine or threonine. But it is able to phosphorylate threonine and tyrosine both, so it is a dual specificity kinase, you can say. Like it has got double specificity. Okay. A very special kinase is V1. It's a dual specificity kinase. Okay. Now what happens is, this kinase can phosphorylate, okay, both serine threonine and tyrosine respectively. Okay. So what happens is, it phosphorylates the mitotic cyclin which is attached with the mitotic CDK. Okay. So you have mitosis. Let me just go down here. So what you have is, you have mitotic cyclin. Okay. that is attached to the CDK molecule, mitotic CDK only. Okay, so CDK1, cyclin B. Now this CDK will contain the one letter code for the amino acid, you can say tyrosine, okay, is Y, threonine is T, okay. So Y means tyrosine here, T means threonine. So threonine at the 14th position and tyrosine at the 15th position is phosphorylated, okay, is phosphorylated respectively by V1 kinase. And once they are phosphorylated, okay, once they are phosphorylated, what happens is the entire cell cycle, okay, gets arrested at the S phase to G2 phase, okay, that cyclin are arrested at S and G2, okay, the transition between S and G2. So what happens is the cyclin CDK complex is not active. Why? Because threonine 14 and tyrosine 15 of the CDK, which CDK here? Talking about CDK1, right? You can write it at least CDK1. Yeah, I have written it here. So CDK1 are phosphorylated by the protein kinase V1. Okay. Now, why is it phosphorylated? Because you don't want the cell to jump from S then G2 to M before the DNA replication even it gets completed. Okay, what you want is the DNA replication to be over, and that too only from one chromosome you should be able to form two chromosome, or from, you can say, all, if you're talking about the humans, the somatic cells, so from 46 DNA you become or you get 92 DNA, not more than that. Means you will get sister chromatids in all chromosomes. You don't want to have three sets of chromosomes, four sets of, you can say, chromatids on each chromosome. So just the double. Okay, so once DNA replication has been completed, okay, only after that will this phosphorylation be inhibited. Okay. Now what will happen is once DNA replication is completed, the phosphatase, just like when we enter the mitosis, the CDC14 got activated here. Once the DNA replication will be completed, okay, what will happen is there will be a specific phosphatase on a CDC25 which will be activated. Now the CDC25, okay, what will happen is it will dephosphorylate the CDK1, and also the active mitotic CDKs will keep on stimulating, you can say, the CDC25, that is an auto feedback loop, and then that will also inhibit V1, okay, by phosphorylating the V1, okay. So what will happen is once you get, so this is what happens, okay, by, if you have CDC25 phosphatase, this mitotic cyclin in cyclin B, yes, CDK1 will be completely dephosphorylated, right? Now mitosis commences. But if you have like this is your V1 that will phosphorylate, mitosis arrested, means mitosis will not commence. Okay, so this is the way that you shift from, you can say, S slash G2 to the M phase. And again, this phosphatase will be active when and only when the DNA replication is over. So again, a great feedback tool. Once DNA replication is over, then only you can even think of going on to commence the mitosis. Otherwise, the DNA replication is not over 1.5 times or 1.7 times it has been able to complete, and you have started the mitosis, then you will be dividing the DNA unequally, right? Two cells will not get equal number of chromosomes then. That is the pathway how you reach the M phase. So now what we have done is we have been able to reach the M phase. Okay. So now we have been able to reach M phase. So now we will be entering the events of mitosis. Okay. So once we enter the events of mitosis, so in mitosis we have multiple phases. Okay. So if we talk about the cell division, so we have multiple phases in cell division. First phase is the prophase. Okay. So there are four major phases. We can subdivide phases also. So four major phases. First I'll explain to you, or first I'll write it down regarding the mitosis. So mitosis is also known as equational division. It occurs in only the somatic cells, means not in testis and sperm, not in germ cell. Okay, not in sperm and ovum. Sorry, the testis would make the sperm and the ovary would produce the ovum. So only it happens in the somatic cells, like your, you can say, intestinal cells, your skin cells, your hair follicular cells. Okay, so only in the somatic cells. Equational division. Why? Because the cells do contain 46 chromosomes, means they are diploid in nature. So what they'll form is they'll form again two cells which are also diploid only. So equational division, number of chromosomes, they stay constant. Okay. Now, now you'll be wondering how 46 gives rise to 46 chromosomes. So what happens in the S phase is, just look at the S phase. We have 46 chromosomes. Now suppose these are the four chromosomes I'm talking about. So this is the first chromosome pair, this is the second chromosome pair, homolog. So what will happen is each one will be able to form its sister chromatid. Okay. And each strand, this strand, if I zoom it in, it is made up of a double stranded DNA like this. And this strand is also made up of a double stranded DNA. So get this concept very clear, how chromosomes are maintained. So these are not single strands, they are each double stranded DNA. So we already have 46 double stranded DNA. Okay. So after the, you can say, sister chromatid formation, these are known as sister chromatids attached to the same centromere. Okay. So sister chromatids, when they are attached to, okay, so how many DNA do they have? Earlier they had 46 chromosome, 46 DNA. Okay. But now they have 46 chromosome but 92 DNA or not. After the S phase, the S phase gets over completely. Now, what would happen is, in one of the phases known as anaphase, suppose each chromosome, like this 46 you are having, first pair, like this 46 total. So now if we divide it from here, so you will get two cells or not? One will have 46. Okay. So one cell like this 46 and another cell also like this 46. Okay. So this is how from 146 chromosome you get 246 chromosomes, because the DNA doubles but the chromosome number stays constant after the S phase. So that's a great 1 mark question. Okay. The chromosome number, it stays constant. Chromosome doesn't become 92 because of it containing the prominent sister chromatids. Only the DNA number becomes doubled. Okay. Then again the DNA number here in a diploid cell, you get 46 chromosome, 46 DNA. Here also 46 chromosome, then 46 DNA. Okay, this is also diploid. This is also diploid. Okay, this is how the equational division or the number of chromosomes are maintained after the division as well in mitosis. Okay, so any doubt regarding this? Ishika, how do the chromosome number maintain themselves, or how does the chromosome number stay maintained after mitosis? And how does the cell enter mitosis in the first place after the DNA replication is over, after we have dephosphorylated the critical tyrosine and the threonine residue, right? So this is how it enters and this is how it maintains the equational division theory in mitosis. It maintains the number of chromosomes which must be equivalent, so that diploid cell gives rise to two diploid cells now. Okay, maintaining the chromosome number or the numerical nature of the chromosome in the somatic cell only. Okay, so any doubt here? Any query in the events of the initial phase of mitosis? Any clarifications needed, Ishika? Okay, good. So now we'll be proceeding to the phases of mitosis. Okay, phases of mitosis. So number one, I'll tell you the broad phases of mitosis. So prophase, metaphase, okay, anaphase, okay, then telophase. These are the four broad phases of mitosis. But if you want to learn it diligently and you can say in depth, via in depth analysis, we can divide it into multiple phases. Okay, we can make it, we can categorize it into prophase, then pro-metaphase. Okay, then we can go to anaphase, then we can go to telophase. After this, nuclear division would happen. Then cytokinesis. So this leads to karyokinesis, the nuclear division. Okay, it's known as karyokinesis. Then after that, we will be having cytokinesis also. Okay, this metaphase I skipped here. Right, metaphase, then anaphase, then telophase. Sixth will be cytokinesis. Okay. So now let's commence our discussion from the specificities concerning the prophase stage. Okay. Now let's begin with prophase. Now each chromosome, we know that is made up of two genetically identical chromatids, what I talked about in the S phase. So now each chromosome has genetically identical chromatids. They are not homologs, they are genetically identical. It's same gene, same sequence. So that is known as, these are known as sister chromatids attached to the same centromere, you can say. Okay. Now, during DNA replication, we know that the genetic material is quite loosely packed as chromatin. Okay. But for mitosis, the DNA needs to be more tightly packed. That is known as something known as condensation of chromosome. You might have heard of this event. Condensation of chromosome. So the thing is, the students, they mug this up completely. Condensation of chromosome, decondensation. But the thing is, they are not aware of why does condensation happen, and first of all, why is it necessary? Why is it important? Why is it of any significance? What would happen? What will be the consequence or ramifications of it not happening? If it did not happen at all, what would be the consequence? What will be the ill effects that could be suffered by the cell itself during the cell division process? The thing is, first of all, I'll tell you something about the histones. Okay, You may not have heard about it, but it is very significant and important to talk about histones with regards to condensation of chromosomes. So histones are the conserved proteins. They are very, very conserved. Conserved in the sense, what does the word conserved mean in biology? This is what conservation of energy we're talking about. Right? We're not talking about M1V1 equal to M2V2 here, recoil of a gun. We're talking about conserved proteins means the sequence, the amino acid sequence of a protein is more than, you can say, 99% identical, okay, in various species. So histones are not found in the prokaryotes except for the Medusa virus, in which the research came out last year that Medusa viruses, being a prokaryote, also contain histones, but they're not usually present in the prokaryotes. So in all the eukaryotes, okay, barring that exception of Medusa virus, in all the eukaryotes, if you see, you talk about annelid, you talk about arthropoda, you talk about human beings, you talk about the plants, the birds, the sequence of the histone proteins, they are proteins, it stays 99.9% identical. Okay, it stays identical. That is the meaning of conserved protein in case of histones. Now, histones are of two types. You have core histone, you have linker histone. Okay. This is your H1, this is your H2A, H2B, then H3, then H4. So these are the types of stones, and there are various variants of each. Right? Earlier it was known that H3 doesn't have any variant, but also research shows that all the stones have specific variants like H3.3, H4.Z, like that. Okay. So the thing is, what histones do is they wrap, the DNA wraps around the histone, and they help to package the DNA, compress the DNA. Because just you think, okay, I'll give you a perspective. If we talk about DNA, okay, we have various types of DNA, A DNA, B DNA, depending on the configuration. So the most common is the B DNA, okay. So depending on the configuration only, okay, don't get perplexed. Depending on the configuration only, we are talking about the bDNA. So once we talk about the bDNA, so bDNA structure, if you talk about, okay, so what happens is the diameter, okay, this is the diameter is 20 angstrom in case of bDNA. Okay. Then the, suppose you have A, T, then G, C. So the distance between two consecutive base pairs in a bDNA, okay, the distance between two consecutive base pairs in a bDNA is 3.4 angstrom. 3.4 angstrom. Okay, and in each turn, okay, this is a turn, one turn, second turn, third turn. In each turn, okay, there are 10.5, or you can say 10 base pairs. So one each turn is 34 angstrom. Okay, one turn is 34 angstrom, means 10 base pair per turn. Really, it is 10.5, but we can take it as 10, round figure. Okay, for simple understanding, it is 10 base pair per turn. Okay, so the thing is, how will you calculate the length of that DNA? Okay, so in a haploid DNA, in a haploid cell, you can say haploid cell, where where we write it as N, means 23 chromosomes are present, like sperm or ovum. Okay, the total length of the DNA can be calculated easily, because what they have is they have 3 billion base pairs. In a diploid DNA 2N, like any somatic cell, okay, like your skin cell only, You have 46 chromosomes. Okay. So number of base pair is 6 billion, 6 into 10 power 9 base pairs. Okay, like this. This is the base pair number 1, this is base pair number 2, like this. So many base pairs are there. Now, how will you calculate the length of the DNA? It is quite easy if you want to calculate. What you can do is, we know the distance between two base pairs and we know the number of base pairs, just multiply it. 3 into 10 to the power minus 10 meter, angstrom is 10 to the power minus 10, again sorry 3.4 it should be 3.4 10 to the power minus 10 meter. Here also 3.4 10 to the power minus 10 meter. This will come till 1.1 meter, this will come till, you can say, 2 meter or 2.1 meter, you can say. Okay, almost. So if we talk about a somatic cell and a skin cell only, what do I mean by this? The DNA present in one of your skin cell is 2 meter long. Just imagine this. Just try to feel this in perspective. In one skin cell, okay, the DNA is 2 meter long. You may not be 2 meter tall. If you are 2 meter tall, means you are more than 6 feet 6 inches tall, right? So more than your entire body length is the length of the DNA of one cell. Now the thing that comes to anyone's mind is, how come the DNA is put inside the nucleus, which is only 10 to 20, you can say, micrometer in length or in diameter. How come the DNA is packaged inside the nucleus? Okay, so the thing is, the histone proteins, they form octamer, means a series of 8. So H2A and H2B, they form a dimer, and the dimerization will be doubled, whereas H3 will form its own, you can say, dimer. H4 will form its independent dimer. So 2 into 2 4 plus 2 6 plus 2 8. So this is total 4 subunits, 2 subunits, 2 subunits. So total 8 subunits. This is known as a histone octamer. Okay. Histone octamer, which is formed by only the core histones. Okay, and what does this octamer do is, this octamer looks like this: 1 2 3 4 5 6 7 8. Now DNA is wrapped around it, okay, like this. We'll go to another octamer: 1 2 3 4 5 6 7 8, like this. And then again wrap around this one. Okay, so DNA is wrapped around this, you can say, 1.67 times. Okay, 1.67 times in a superhelical fashion, left-handed superhelical fashion. Okay, and the number of DNA base pair contained in one nucleosome octamer, or sorry, one histone octamer, is 146 to 148 base pairs, and this entire thing is known as a nucleosome. Okay, and from here to here is almost 200 base pair. From here to here it is known as a chromatosome. And the DNA linking the two, you can say, histone octamer, this DNA is known as linker DNA, and this is bound by linker histone H1. Linker histone H1. Okay, this is the first level of compression. Then you will be having the beats on a string theory, and also I'll just show you the structure of nucleosome. You will be amazed to see how the DNA is compressed. Just see, look at this structure. Okay, this is the Nature paper. This is the most beautiful diagram in biology. Just look here. What is the difference between a chromosome, a chromatin, okay, and DNA? One should know, or one should be aware as a biologist, the real difference between a chromosome, a chromatin, and a DNA. Free DNA is this one, 20 angstrom, so it is 2 nm. Okay, so free DNA is this one. Now, what happens is the DNA is compressed, and first nucleosome is produced. This is one histone octamer. You can see here the diameter of the DNA is 11 nanometer. Okay, 11 nanometer diameter. Now, again, it has been, you can see the twists. More and more twists are being put on the same histone octamer. The diameter is becoming 30 nm. Okay, 30 nm fiber from 11 nm, you are becoming 30 nm. But more and more twists, so more and more nucleosome are put in 30 nm than you can imagine in 11 nm. More and more twists again. So now the diameter is 300 nm. Okay, then again see more and more twists, 250. Okay, so then we have 700, and this is the chromosome 1400 nm. Entire DNA is put here. Okay, entire DNA, okay, is put here completely. Okay, so this is the chromosome, the chromatids of the chromosome. You can see this structure. Okay, so the thing is, we have beads on a string model, this one. Okay, then we have the solenoid model, this one. Then we keep on, you can say, compressing. So this model has not been elucidated directly in research, okay, but there have been indirect evidences of this happening. Okay, so the thing is, only till beats on a string model has been, till this model only, nucleosome and maximum till this has been elucidated in research papers. Okay, directly through the cryo-electron microscopy, they have been able to elucidate. So this is the level of, you can say, compression. Okay, so this is chromatin. Okay, this is your chromatin. The 300 nm one is the chromatin. This is the chromosome. Okay, this is the DNA. You see the difference. This 300 nm is the chromatin one. Okay, just get the difference. 300 nm one is the chromatin one. It is still haphazard, but this is really condensed. 1400 nm one. That is the chromosome. The chromatids of the chromosome. This is chromatin. This is chromatid of the chromosome. This is simple DNA. Okay, the level of compression. So now tell me, is it clear, Ishika, completely? How does this happen? The compression of the DNA, how did I calculate the length of the DNA and all? And why do we need to compress the DNA? Because it could be 2 meter, otherwise where will you keep your entire DNA, right? Of 37.3 trillion cells that we have in our body. Okay, so is it clear till here? Compression thing? Any doubts in DNA compression and the difference between the chromosome, the chromatin, and the DNA, free DNA? So those three things are not the same. If I say chromatin, chromatin means 300 nm. If I say chromosome, yeah, 400 nm. Sorry, 1400 nm. Yeah, good Ishika. And then if I say DNA, that is the 20 angstrom one. Okay. So now come to the understanding of why chromosome condensation is needed in case of prophase. Because if we go for division of chromosome, okay, so what are we doing here? We expect this structure, okay, then we can divide it into two equal parts or not. Then we can divide it into part 1, part 2. Okay, later on we can do like this. But if we have like this is the chromatin stage. How will you divide it into two equal parts? It will get tangled up if you just break it down. How will you break it into two parts? There is no centromere region here, no real. How will you divide? From where will you break? It will get all tangled up, right? Because it's like a thread, a completely folded thread. So it will be very difficult to divide it into two equal parts. It may get tangled up, broken in, you can say, wrong places, wrong locus, and then you will be having unequal distribution of DNA. So what we need to do is the DNA is present in the chromatin form in the cell, chromatin form in the cell. But when the cell reaches the M phase of the cell cycle, it has to be condensed from 300 nm1 to the 1400 nm1. That is the chromatid of the chromosome, or simply you can call it chromosome. Now, when we have condensed it, then we are getting a very clear cut picture, a clear cut dissemination of how to divide the DNA equally, because you can never divide the DNA equally. You will produce tangles, will produce breakage at the wrong places. Now here you have the centromere also. Here the kinetic or microtubules arising from the centrosome, which contains centrioles in almost the perpendicular fashion, will join here and will pull from both the sides. Okay, later on in the next class I'll show you, it will pull, and then the two chromosomes will be divided, the sister chromatids will be divided equally. Now both the cells will get equal number of DNA, which is impossible from the chromatin stage if you are in the chromatin stage. That is why we go for condensation. This process is known as condensation because we are condensing, we are further compressing the DNA. This is known as condensation of DNA. That is the first point pertaining to the prophase stage, condensation of DNA. That is the reason nobody tells you. Okay, but this is the reason why you should be knowing why condensation is happening, because biology is all about knowing the reason, understanding rather than knowing the facts, right? So this is why condensation of DNA is required. Okay, so tell me, Ishika, was today's lecture completely clear? You had any kind of queries, any kind of doubts, any kind of clarifications needed? Any kind of queries remaining, that why is condensation important, why is condensation required, what would happen if condensation does not occur, what will be the ramifications of it not happening? Okay, so since the DNA stays in the chromatin form till the, you can say, G0 G2 stage, you have to compress it in the M phase in order for the DNA to be divided, segregated in two equal identical numerical ways, right? Okay. So in order to maintain the numerical equality in both the daughter cells, the DNA needs to be condensed. Okay, Shika. Okay, so stay safe, stay healthy, have a nice day, and thanks a lot, and we'll meet in the next class. Okay, Shika, bye.